Obviously, you've got, Punnett squares and stuff that I'm not putting up here, but the answers, I'm just going to give you the answers so that you see if there's something wrong with what you're doing or not. That's what your answers ought to look like for 1 through 5. If I was grading this, you wouldn't get credit unless I saw Punnett squares that were correct. But for you to kind of just know if you're on the right track or not, do a quick check here. Make sure you're on where you should be. One of the hardest things about problems like this is sometimes you forget to actually answer the question. Some of you probably have some of these where you got the Punnett square exactly right. You just got to answer the precise thing they were asking. So that's what these are. For number 5, this means Sniffles, Esmeralda, and Whiskers. Those are the names of the mice. Any questions at this point? Anything that does not add up? Anything you think I might have screwed up? Because that's totally possible. I typed this very fast this morning. Nicholas? Everything look good? Yeah. Okay. You're the one I know is going to catch it if I mess it up.
For number 1, could it be possible to use lowercase?
No, because we know that both, wait, was it groundhog? What were they? Guinea pigs? It can't because they're both black. If one of them was lowercase, they'd be white. Right? So it's a cross between 2 black guinea pigs produces some black and some white. So it has to be 2 heterozygotes. And honestly, that model of that problem, where you see 2 dominant showing parents, and we want to know what their genotypes are, throughout the entire chapter, you're going to be playing this same game, which is, do they have any recessive kids? Because if they do, we know what they both are. That's the game, and it doesn't change a whole lot. It gets more complicated. You're still often just, if they're dominant showing, you're looking for the recessive kid. Other questions? Okay.
2B, please.
2B? Sure. For number 2, what letter did you pick?
Number 2. B.
Letter B? And big B was yellow, and little b was purple, correct? Okay, and so if we're talking about the F1 plants, these are the ones that are all heterozygote. So it's going to look like that, right? And so we're going to get that ratio phenotypically that is basically 3 of them showing dominant and 1 showing recessive. Does that make sense so far? And so all they're doing is saying, okay, well, let's say we grew 600 babies from this cross. How many of them would have purple flowers? Well, that's this, which should be what fraction of those 600?
1/4.
1/4. So that's all the math you had to do, is just divide 600 by 1/4 by 4, which is 150. Good? Okay. Let's see what happens when we look at 2 traits at once. Grab your notes and grab a blank piece of paper, please. Then next question. After discovering dominance and recessiveness and segregation, his next question was, well, what if I start to cross pea plants from different gardens? Yeah, okay, this guy before me kept purple and white flower plants in separate gardens. And then he kept tall and short in another pair of gardens, right? And then he messed with pea pods in another place. What if I start to look at more than one trait at once? What if I look at flower color as well as plant height in the same plants? And the question he had was, do they influence each other? Do these different traits change each other's probability? And what he came up with, and he was right for the genes he picked. We will find out that he was not completely correct in what he assumed. But what he saw with the traits he was looking at, and what we're going to see with these traits we initially look at, is no, they don't. They don't affect each other. They assort independently of each other. In other words, let's say we're talking about hair color and eye color, and your parents are heterozygous for both. Just because you got the dominant hair color allele does not have any influence on whether you get the dominant eye color. They assort independently. They do not affect each other. Which means if we are tracking two genes at once, we're dealing with a wider range of probabilities. We call this a two-trait cross, and it results in a maximum of 4 possible gametes each parent can make. And I always kind of just like to see how many of you can puzzle this out for yourselves. It's usually more than I expect. So let's try one. Let's try a two-trait cross without any other instructions from me. Grab that blank sheet of paper you got. Let's keep the letters easy. And let's say that we got, and remember, it's a good idea to just kind of write these down. Green. The big B is brown hair, little b is blonde hair, big T is tall, little t is short. By the way, both of these genes are far more complicated in real human beings. We're just oversimplifying here. And let's just say we are mating 2 heterozygotes for both traits. So that's a big little crossed with a big little. See what you can do based on what you just wrote down, which is that they do not affect each other. And that when we track 2 traits at once, there's a maximum of 4 gametes from our eggs each parent can make. See what you can do. See if you can set up the Punnett square. Many of you have already figured out a key bit of information, which is that a Punnett square has to be bigger now. We're not just looking at 2 possible gametes from each parent. We're looking at 4, which means this, right? 4 by 4. second thing is the really important one. So far, I've seen 3 of you that have absolutely nailed this. You know what's going on. The rest of you are making the key mistake that everybody makes. You're just putting one letter over each column. You're going, okay, well, I'll put a big B here, a big T here, a little b here. That's going to make a half a kid. That's not going to work. Doesn't a sperm or egg have to have one of each? Doesn't a sperm or egg have to have hair color and height on it? Otherwise, your sperm is going to make a kid who has brown hair and 0 height. That's not a kid, right? You got to have both. So what you are really doing is you're determining how many different ways this parent can put a B and a T together. And 3 of you already know. The rest of you just kind of need to play mix and match here. And go, okay, well, what's the first possibility? That, let's call this dad. What's the first way that dad can put a B and a T together? Ella? How about a big B and a big T? I like it. What's another one? Will?
A big T and a little T.
Big T and a little T, that's a tall kid who with no hair.
B, T.
Little B, little T? Sure, that's one. I always put that last. What else you got? Big B, little T. What's left? What should I put here?
Little B, big T.
Yeah. There. What goes on the left? The other parent is exactly the same genotype, so same thing, right? Good. The only other thing you need to know is that when we put kids in the squares, we want to keep them like their parents. In other words, we want BBTT, not BTBT. Right? We want to keep the B's together and the T's together. We want to keep the capital B before the lowercase b if it's heterozygous, and same with the T's, so that we can read them just like their parents and tell easily what phenotype they are. I'll let you work. Why don't you, now that you know how to do it, go ahead and put it in your notes. Put this big old Punnett square in there. Remembering what we're doing here. We are determining possible sperm and eggs and how they can join to form kids. If you are still swimming a little bit here, if you don't understand why we set up the top and the left side of this square the way we did, please watch carefully. For that first left square, we went big B, big T, because that's one way that that parent can do that. Then we went big B, little t, because that's another way we can put them together. Then we went little b, big T, and then we took the 2 littles. And we did exactly the same thing down the left-hand side. And now we're just starting to put together BBTT, BBTT from the top and the left. I hope.
From one trait, would there be less possible?
As soon as anybody's homozygous for anything, good question, your Punnett square can get small. This is the biggest Punnett square you're going to have to make. Because they're heterozygous for both, so they have the maximum possibilities for the 2 sets of gametes, right? As soon as anybody's homozygous, you got less possibilities. Your Punnett square can shrink if you really know what's going on. So finish it up, make sure it matches mine. Then we have to actually answer the question, because we haven't yet. When you're ready, start counting. How many of these kids, because we want a phenotypic ratio here, how many of these kids are going to have brown hair and be tall? In other words, how many kids are going to show dominant? Go ahead and circle those. Circle the dominant showing kids for both. Brown hair and tall. How many are we looking at? You got a number we're confident in? Ella?
9.
9. Good. So brown hair and tall, we have 1, 2, 3, 4, 5, 6, 7, 8, 9. If you set it up the way I did, it's a nice little triangle. 9. How about brown haired and short? Let's put a, I don't know, put a diamond shape around them. Mark them as different. Put a diamond around them. So that's going to be at least one big B, but 2 little t's for that second one. Make Colby. 3. 1, 2, 3. Now we want blonde hair and tall. It's nice that we've marked these out, right? Now you know exactly what you have left. There's only 4 left. How many of them are blonde haired and tall? 3. 1, 2, 3. Just got to mark them differently somehow. It can be dots and lines, whatever you want. And then finally, our double recessive kid, blonde and short, for a total phenotypic ratio of 9, 3 to 3 to 1. Dominant, dominant, to dominant recessive, to recessive dominant, to double recessive. On your way out, pick up the homework, which is numbers 1 through 5, the front side of this, not the back side. 2 day crosses, due tomorrow. Review stacks right here. You should take 1. 1 through 5. Just the front side. I think it spills onto the back just a touch. Just a couple of words.